Our keynote speaker today is actually one of my favorite scientists. I'll just get it out there so you know. He's the chair of the Department of Immunology and Microbiology at the Scripps Institute in La Jolla. They made him a name chair, and he's also the director of the Consortium for HIV/AIDS Vaccine Development and scientific director of the Neutralizing Antibodies Consortium for IAVI. Now, it's really fun to get someone who's so accomplished to get their CV because you get to see what they did before HIV. So Dennis was trained as a chemist at Oxford and then did his PhD in Sweden, and the title of his thesis was Metal Ions as NMR Probes in Biology. So you can see that he started one place and wound up in quite another place. He came back to Oxford and where he had the title of extraordinary lecturer. I'm sure there's a good story there that we'll get to hear sometime, but not many of us get to have that title. And then as a reader of CVs, I would, if this was my CV, I would interpret it as he got his real first job at the University of Sheffield, where he stayed for about 10 years, but they made a terrible mistake. In spite of promoting him and giving him a chair position, they let him go to Scripps as a visiting scientist. And so in 1991, he actually moved to Scripps as a professor, and he's been there ever since, and he's been chair of the Immunology and Microbiology Department since 2015. He's lots of awards, and the one that would resonate with all of us, though, is one that comes through CROI, is the Bernard Fields Award. So Dennis got to start in looking at NMR and protein, DNA and protein using NMR. But even early on, there was this interest in IgG that's followed him through his whole life in various forms. And I guess I first became aware of Dennis as he moved more into the HIV field. And what he brought to the field was these innovative ways of thinking about generating antibody diversity. And kind of a real watershed moment in the field was this protein, or this antibody that he literally made, called B12, and I'm sure we'll hear about B12 on the way. And Dennis, I hope you also tell us the secret of how immunologists name antibodies, why it's called B12. I think it's fun to know that answer. One other thing that's a feature of Dennis's work, and he's been amazingly productive, and I had to go all the way back to find Aesop that said this, and it was a man is known by the company he keeps. So Dennis has surrounded himself with an amazing group of scientists at Scripps, and they really are setting the pace both in the science and in the thought of developing antibodies against HIV and vaccine approaches. So Dennis, thank you very much for coming today.
All right. Okay, can you see the introductory slide? Is that?
Yeah, you're good.
I'm good. Okay, thank you. Thanks very much, Ron. Very kind words. Very kind. So I'm here in La Jolla, in lockdown, a long, long way from Cold Spring Harbor. I actually have fond memories of Cold Spring Harbor. I taught a course there on phage display for a few years with Carlos Barbas. And then I organized later a meeting on vaccines. So I spent quite a lot of time there, and it's always a great place to go to. It's kind of nice to do something on HIV. I must say the last 2 months I've been totally immersed in SARS-CoV-2, which I'll say a little bit at the end. So it's nice to go back to HIV. And Ron and Zandri asked me to give a talk with historical aspects, so where broadly neutralizing antibodies came from. And it's kind of fun, and it made me go back and look at some very, very old slides. And but I think there's a great danger here, which is, history can be pretty boring actually at times, I mean, honestly. And you don't want to hear about how we had to, you know, wash our own test tubes and that kind of stuff. So I wanted to make this a little bit different. So I thought I would go back and bring us from a long time ago to the now, but focus on what we got right and what we got wrong, and what we still don't know about. And I'll kind of keep coming back to that. And also some general thoughts about basic concepts. And not all of these everyone agrees on by any means, but I'll give you my kind of take on some of those. And I think, you know, what's happened with HIV in the time, at least I've been working on it, I think when it started out, it was sort of behind, you know, HIV people used to do and say things that were pretty naive in terms of immunology and vaccinology, but I think that's slowly reversed. So now, at least in the antibody field and in vaccinology, HIV is actually leading the way in some respects, and I think that's kind of interesting to have seen that. So, having said all that, I'll go back and so let me see. So let me begin with neutralization and protection, which is where it all started for us in terms of antibodies. And so what I did early on was to, when I went to Scripps, was to work on libraries, antibody libraries. Mouse monoclonals could have easily be generated with the Kerr-Milstein approach, but human antibodies not. And the new library approaches offered a route into human antibodies that was much more reliable, and we chose to apply that to HIV. And so this is work that that really got us going with Carlos Barbas, who unfortunately passed about 6 years ago now. And we isolated this antibody from an infected donor, and the antibody was cross-reactive, cross-neutralizing, broadly neutralizing. And this was a new concept at the time, and people didn't necessarily think this was going to be possible because they looked at HIV, they looked at how enormously variable it was, and said, well, how can one antibody recognize so many different isolates? And so, and the way we got it was basically, we had a donor infected with one virus, obviously, and we selected with an envelope protein of a different virus. So, you know, you're going to select for cross-reactivity. And we got this antibody, which is called B12. And I'll tell you now, various people have thought that was something to do with my name. It is not. The B12 refers to the fact I used to call it 3B12 because we pulled it down with GP120 from the 3B strain, and then I just got rid of the 3. And so that was as simple as that. And that's why it's a small b. And it wasn't clone number 12. And given that at the time, you know, some skepticism around breadth of neutralization of primary isolates now, I actually had this tested at 3 different labs listed and 3 different assays just to be sure it was right. And it was, and it came up good in all of these assays. And so we concluded it was okay. And it then was used very widely along with Herman Kattinger's antibodies that he isolated at a similar time. So that was neutralization. So we now had a broadly neutralizing antibody. And so we wanted to know if it had protective activity. And that was difficult. In those days, we went from a Hugh PBL SCID mouse with this T cell. Can you see my cursor? Yes.
Yeah.
Good. So this is a T cell line adapted virus. We did this with Rick Kaup. And what you'll see is that if you look at the curves of neutralization against concentration here is the the neutralization, and then the protection is actually requiring 2 logs higher concentration. And that was a theme we established first there. I actually think that's a very common theme in virology, that if you seek to protect, you need concentrations that are pretty strong multiples of the viral load, of the neutralizing titer. And you can see that there. I'll come back to that. And then we did it with, in this model, with primary viruses. For primary viruses, you needed more. You see the doses here are higher than against the lab adapted, much harder to neutralize. The harder the virus was to neutralize, the more antibody you needed. And the antibodies, the antibody was ineffective after a given time. So there was a bit of prophylaxis here, pre-exposure prophylaxis, but not much. And very rapidly, the antibody became ineffective. And we then, this was the days of T cells, and still the days of T cells, don't want to say that, but the days of T cells dominating HIV vaccinology. And it was hard for us to get monkeys to do experiments on, for example. Here we got a very limited number. And because of the very good relationship between neutralization and protection, using only 4 animals here per group, which people would have a heart attack over these days, you could still pretty much see what the result was. So, this is a vaginal high dose challenge with the SHIV, so B12 and HIV antibody protecting. And you can see that at the high concentration, high neut titers, complete protection, intermediate titer, intermediate protection, 2 protected, 2 not, and low, no animals protected. And pretty good, and this I think was the first titration of protection. And it again fits with this notion that you need, or this observation that you need a lot of antibody on board to provide sterilizing immunity. And we then got a fairly clear, over a number of viruses, good relationship between neutralization and protection. And that's 2001. And that's actually now being borne out. This is recent paper with John Mascola in just looking at macaque data. And you've still got this, it's slightly different plot, but you've still got this pretty good relationship between neut and protection. And you've got this multiplier here. So about 100-fold higher serum neuts to provide protection. So we got that right. And I think that's been consistently seen even down, people really didn't like that at the time, the Hugh PBL skid mouse. That's pretty damn crude. It is. But it's been borne out many times, up to at least MHPs. We'll now see the ultimate test here now is the AMP trial, which is the VRC01, broadly neutralizing antibody in about 1,000 humans. See how that protects. Pre-exposure exposure prophylaxis study. We'll see how that protects, and the results of that will be out in the fall of this year. So we'll see if that story goes all the way from the very small, crude mouse studies out to humans. With this antibody, we did all sorts of things. We were very interested to look at the role of effector function in antibody protection, which actually hadn't been done. It had been done a little bit in the 80s, particularly, not with HIV, but with various viruses. A little bit actually by particularly Mike Buchmeyer did a fair amount of that at Scripps, and then it had not been looked at. And actually recently you'll see a lot of papers on the role of Fc receptor in virus protection, but here it was not so looked at. And we chose to investigate it through mutations in the antibody, the B12 antibody structure. So we used single point mutations to eliminate complement, and then a pair of mutations, so-called LALA mutations, to eliminate FCR binding. And when we did that, and I'm not going to bore you with all the data here, but when we did that, knocking out complement didn't change protective ability in macaques. But if you also knocked out FC receptor binding, then now you did have an impact, and about half the animals now were protected under the same conditions. So a clear contribution of FC receptor binding to protection for this antibody. We did more studies. It didn't look like ADCC. We've not been able to pin down exactly what. But then, and this is where I would say we've got some, we didn't get it wrong. I think we got it right, but it's more complicated. So we have now many more potent antibodies, and I'll come back to that. But if we consider now the potent antibody PGT121, then we find that this, and we've done a lot of experiments on this recently to try and nail it down in all sorts of complexities, but the conclusion's pretty stark that protection is not augmented by FCR-mediated effector functions. So for an antibody that's much more potent at neutralization, there does not seem to be any involvement of FCR-mediated effector functions. It seems only if you've got a less potent antibody. And there are possible reasons for that, including the fact that with the less potent antibody, you have a lot more antibody on board. You may have a lot more antibody bound to various FC receptor bearing cells and so on and so on. But I would say that was an example of where, you know, there's some lesson there that, you know, antibodies don't care. They can work however they need to work. And, you know, here's a clear difference between two antibodies. So a complete generalization would not be correct. And then I wanted to put this slide in briefly to just make a couple of remarks about neutralization and protection, because to me this is often oversimplified or even misunderstood. So some folks say, you know, well, it's protecting by neutralization in vivo, and actually they very often don't know that. And I would like, and I think a number of people over time have kept to this, that neutralization should be reserved for the in vitro loss of infectivity that you observe when antibody binds to the virus. So it's an in vitro assay, and you can clearly measure it and define it, and that's neutralization. However, that neutralization is only possible if the antibody binds to functional structures, functional spikes on the virions, and you know, then therefore will bind to infected cells. So in a sense, it provides a readout of binding to functional structures. And so if it does that, then in vivo, it's true, you could be using neutralization to as an in vivo mechanism of protection. But it could also be anything that correlates with binding to functional spikes. So, for example, it could be ADCC of infected cells, or phagocytosis, or anything that required binding to functional structures. So it's hard to actually say that the mechanism that's operating in vivo is precisely neutralization. There are very few examples of where that's been shown. And in some ways, I think the best way to think of a neutralizing antibody is one which binds to functional structures on the surface of virions and infected cells. And in vitro, if you bind enough, that leads to neutralization. In vivo, it could also lead to neutralization, but there are other possibilities as well, like ADCC or ADCP or whatever. So you could imagine that you you've not bound enough antibody to the virus to neutralize it, but it was still enough to have the virus taken away by a phagocyte or something. And I think that's, you know, just worth remembering. And this is all applies to neutralizing antibodies. Non-neutralizing antibodies is a different story. And then also on this sort of general theme about thinking about neutralization, neutralizing antibodies and so on, what I've often seen, and even more recently, is representations of neutralization that bear no relationship to the sizes of the molecules that are actually involved in the neutralizing process. So what we have here is HIV entry, and you have the spike, the heterotrimer GP12041 shown here, and you have the two receptor molecules CD4 and CCR5 more or less to scale. And what neutralizing antibodies are going to do is to bind to the spike, and this is how big they are relative to the spike. And it turns out that for HIV at least, the binding of a spike, the binding of an antibody to a spike is necessary and sufficient for neutralization. And, you know, there's beautiful experiments done by Sadrowski and DeRosier, for example, and us, that show that you can put a peptide wherever you like, a peptide epitope around the spike, and then you can take an antipeptide antibody and it will neutralize. And we presume it does it because of its bulk. You know, it'll either inhibit attachment or inhibit the fusion process. So I think that's kind of important to always think about the, you know, the relative molecular sizes of these things. And then, and this is one thing that comes up quite a lot, and that you don't make, or I don't make myself popular with, which is the structural people. They like to get a structure, a recombinant molecule of an envelope spike, and then they bind an antibody and they observe some phenomenon, and then they say that's the mechanism of neutralization. Well, it might be, or it might not, and because neutralization involves the binding of antibodies to usually to an array of spikes interacting with an array of receptors, and you know, the rules could be different. And so, you know, if you take SARS-CoV-2 recently, there's a paper on bioRxiv that says, you know, the antibody is an antibody and it doesn't bind exactly to the footprint of ACE2, and it doesn't inhibit soluble ACE2 binding to soluble RBD on the S protein, and therefore the mechanism of neutralization is not inhibition of attachment. Well, that may well be true, but it may not. It may be that once you get an array of these things binding together, that the proximity of the antibody to the ACE2 site is sufficient to inhibit the binding of ACE2 when it's stuck on a membrane in a multivalent format. So I'm just pretty cautious about these, and I argue with the structural people, and you know, they observe their changes and they like to think that that's the end story, but I really don't think it is. So that's neutralization protection. So what we were also interested in was therapy, and we took Pascal Poignard took a shot at this 1999, and again we used a very simple model, the Hugh-PBL SCID mouse model. So we simply populated mice with human PBLs, and then we infected, and the infection goes. Eventually it burns out, but it runs for quite a while. And we let it go for 11 days, and then we administered a cocktail of 3 neutralizing antibodies, including B12, and then we looked to see what had happened. And remember, we infected with a molecular clone, and we only let the infection go for 11 days, and then we did our treatment. And lo and behold, at least in one of the mice when we did this treatment, we isolated triple escape variants after such a short time. And that made us pretty skeptical about antibodies in terms of therapy. And I think generally around that time, there was pretty much a feeling that, you know, antibodies are good for prophylaxis, but maybe not so good for therapy once infection's going. And I think we were wrong on this, and not exactly wrong, but the data's all fine. But, you know, this is with a certain potency of antibodies. When you take the potency up, you know, 2 logs, then you start to play a different game. And that's exactly what happened in this paper that we did with Dan Baruch, where we took one of our more potent antibodies, actually the PGT121 I already mentioned, and we treated monkeys. And for some of the monkeys, there was a rebound, but for the animals that came in with a relatively low viral load, when we treated them with monoclonal antibody, they had sustained lowering of, sustained disappearance, really, sustained control of virus, even out much longer than beyond when the antibody had long since decayed. And, you know, the only thing, and what seems to be happening is that you get the intervention with the antibody seems to allow for some resetting of the cellular immune response, and you can gain control under certain conditions. And that's been borne out in a number of different studies. And these antibodies are now, you know, going into humans for therapeutic use, and particularly by Michel, for example, we also, and Dan Baruch and the VRC. So, you know, the increased potency has changed our views on the therapeutic potential of these antibodies. Okay, so that's the antibodies themselves. You get more now onto envelope structure and an HIV vaccine. And this is something that I wrote in 1997, which I actually went back to look at for this talk. And it's kind of interesting, and I'm surprised actually at, I don't generally like to go back and look at too many of these things, because you do get quite a few things off kilter, but this is not bad. And it says antibodies that bind well to envelope can offer protection or benefit. The challenges for vaccine design are to elicit such antibodies involved in transmission in humans, primary viruses. 2 major obstacles exist. The first is very little of the envelope spike surface appears accessible for antibody binding, low antigenicity, probably because of oligomerization of the constituent proteins and a high degree of glycosylation of one of the proteins. Remember, this is before there's any structures. There's not even a structure of GP120 yet. The second is the mature oligomer constituting the spikes appears to stimulate only weak antibody responses, low immunogenicity. And those 2 things I think are really borne out now. This one is not. So viral variation is another possible obstacle that appears to present fewer problems than anticipated. I think that's understated. I think the variation through immunodominance causes a lot of problems, reflecting back on the immunogenicity here. Vaccine design should focus on presentation of an intact mature oligomer. We do that. Increasing the immunogenicity, we do that. And learning from antibodies available that potently neutralize primary viruses. Those are all actually still very active, I would think. Okay, and I've just included from that review a model that we made prior to structure. And always if you make models, you know, you kind of, people make fun of you in some ways, structural people particularly, and they're perfectly entitled to. But, you know, I still think they're very helpful. I thought this was very helpful, and it still presents a lot of the broad picture, actually. You know, it's difficult getting into that CD4 binding site and these other places. And these parts of these loops, the V3 in particular, it is out there on the lab-adapted viruses, and on the primary viruses, it's not. And And a lot of this is down to work by many, many different labs. I'm saying the models are a result of you know, the work of many, many different people, that John Moore, Joe Sadrowski, and all sorts of folks that allowed a building up of a picture, and ourselves. But nevertheless, what you really want is real structures. Erica Sapphire we've been working with Ian Wilson for many years on this problem. Erica Sapphire was a student in Ian's lab, PhD student. She decided to try and solve the structure of B12, the FAB, couldn't do it. And then eventually she said, well, I'm just going to try for the whole antibody. And that just fell out. And she solved the first structure of a whole human antibody. And that was actually B12. And that's what it looks like. It's not quite as nice and symmetrical a Y as you often see, but that's actually a real antibody. And when you looked at the Fab, you found this long H3, like a finger out here. Here's the H2, heavy and light chain. So we modeled that, and the modeling looked really good. Now we have the structure of GP120 from Peter Kwong, and we modeled it, and we put the CDRH3 right into the CD4 binding site, and that looked great. The modeling looked great, and it was wrong. Actually, it's the CDRH2 that goes into the CD4 binding site. The CDR3 goes along the side. So we actually got that part of the modeling wrong. Peter, in his structure, 2007, you know, that was a definitive structure, but he said there's no, this is the VH, the VL's behind here. There's no involvement of the VL. It's purely a heavy chain interaction. Now, we had mutational data that suggested there was a region of the light chain, an arginine shelf here, that was very crucial for binding. So we couldn't understand that. And then eventually, years later, the structure was solved with a full envelope molecule. And the answer is yes, you do need the light chain. It interacts with the glycans. So this structure isn't completely right either. So, you know, there's layers here going forward, and now we have a pretty clear picture. So our mutational data was actually correct. Okay, but as we were doing all of this and doing this antibody work and moving more to getting structures and bound to GP120 and so on, it became increasingly apparent that the classical approaches to vaccine development weren't going to work. You weren't going to just immunize with GP120 and get an HIV vaccine. And we argued here for the first time, I believe, that we had to go and look beyond the molecules of natural infection. And that is exactly what has happened. So, you know, I'll show you in a second that, you know, one of the key molecules involved in trying to get to an HIV vaccine is you know, far removed from what you call a molecule of natural infection. So that was important. And bringing these things together, so I tried to see how, you know, what would be a vaccine strategy for HIV in 2002, and came up with this that's been termed by Reno reverse vaccinology 2.0. And the basic notion you've probably already or seen or gathered from what I said is that we would isolate these broadly neutralizing antibodies. We'd investigate them at the molecular level, determine the shapes recognized, and use those shapes as a vaccine. And so we're working backwards from good antibodies to a vaccine. And actually, this approach has yielded quite a lot, not for HIV yet, but Peter had done beautiful work to come up with, and Barney Graham, to come up with an RSV vaccine that's based on first finding an antibody that recognizes the pre-fusion form of the F protein. Antonio Lanzavecchia defined a new epitope, the pentavalent molecule that's driving CMV vaccine development. And universal flu really comes because people have broadly neutralizing antibodies to flu. And that's the that's their driving force. Okay, so hope I'm still okay here. What we knew was that we needed better HIV antibodies, and in order to do the sorts of reverse vaccinology and so on, the antibodies weren't good enough, and it was the coming of high throughput technologies that really changed things. And I'll just show this in a couple of slides. So, and this has a big lesson, at least for me. So what we wanted to do was look at the cross neutralization. So you have a list of antibodies here, you have a list of viruses down here, and you have a color-coded ability to neutralize. So if it's blue, it's not doing much. If it's red, it's neutralizing. And what we wanted to do was look at this on a much grander scale. And so we worked with Monogram, and this was robotic screening now. And otherwise, you know, somebody would have had to have done God knows how many thousands of measurements here, because you're titrating each of these, right? So it's not just a single. Each time you're calculating a half K50 or ID50. So you know every one of these has got a titration in it, and so it's huge. And robotics is the way to do it. And they don't, at least in 2004, you know, you couldn't get robotics money and to do this kind of study. Where's the hypothesis-driven research in this on an R01? And the way we did it was we'd linked up with IARVI, we'd formed the Neutralizing Antibody Consortium, and IARVI had money, and they could afford to spend this money on this kind of effort, which we could have never done otherwise. And this, you know, allowed us for the first time, and this was the first time this was done, to look at a large panel of antibodies against a large panel of viruses. And we could see B12 was good, particularly against clade B, but it weren't so good against some of these other clades. And we knew we needed better antibodies. So, and this was really, Wayne Koff was absolutely instrumental here, the CSO at IARVI. So what we said was, we need to look through lots and lots of donors, find those with the best neutralizing antibodies, broad neutralizing, get them, and then try and isolate monoclonal antibodies from them. And so and this was, again, you know, you can't do this, you know, on an R01. I mean, IARVI had all these clinical trial sites. They pulled 2,000 donors from across the world. They had certain criteria that was, you know, that they had been infected for a long time. We knew that was helped to develop breadth, and that they weren't on antiretrovirals, and they had good CD4 counts. And we found 2,000 of those, and we shipped them all to London, and then we shipped them to Monogram. We had the high throughput set up now. We could screen large numbers of sera against large numbers of viruses. We did all that. Eventually, we reduced the number of viruses to an indicator panel. And now we found these donors that we termed elite neutralizers, who neutralized very broadly. So, you know, they had titers in the thousands against very different viruses from different clades. So we said, these are the people we want to study and try and find the monoclonals doing this job. And then we hitched up again and again. This is dependent on having, you know, access to further funds. We hitched up with a company that were then called Spalterdac, that I'd been asked to look at because they were looking at antibodies to in tumors, and they had this ability to do single cell cloning. And so I said, well, you know, this could be great for HIV. So we hooked up with this company. We took a donor. They plated out the B cells from one of these elite neutralizers at about 1 B cell per well, activated, and then we had enough antibody made from single B cells to do high throughput neutralization screening with Monogram. And, you know, out of thousands or 30,000 antibodies, we found 2 that were really good. And here they are, you know, now potency is coming down. These antibodies are neutralizing lots more viruses at much higher potency than, say, B12 that we've been working with. And you can see that on here. So there's B12, global coverage, potency, and here's this new antibody, PG9, nearly 80% of viruses very potent, relatively very potent. So this, you know, this upped the ante hugely, and then we got more and more of these antibodies, ever better. Others joined, the VRC, Michel Nussensweig, others since joined, and now there's, you know, huge numbers of these very potent, very broad antibodies. We term these days super antibodies, and the key to finding these antibodies is, number one, look at a lot of donors, number two, mine their repertoires very deeply. And so I think, in part, you know, the view that, you know, antibodies only, let's say, you know, I was brought up with like lysozyme, there's like 3 or 4 epitopes and that's it. I don't think it's true. I think antibodies as any part of any protein, if you just look deep enough, you know, they have the capability to get into every nook and cranny on every protein you've ever thought of, but maybe only 1 person in 1,000 does it, and maybe it's 0.01% of their repertoire, you know, so, but they're there, and, you know, increasingly people are finding them. Okay, so I'm getting towards the end. Envelope trimer structure. How long do I have, Ron?
You know, Dennis, you're welcome to go as long as you want.
Okay.
Nothing planned afterwards, so
Okay.
I'm enjoying it. Please go on.
Okay, thanks. Okay, so envelope trimer structure. So, you know, you saw my kind of crude models. Things really took off when John Moore, Roger Sanders, James Binley, Andrew Ward, Ian Wilson, in a series, you know, it was a long time for them to put all this together, but, you know, they generated and solved the structure of bona fide recombinant trimers that looked a lot like the native trimer on the surface of virions. And this, you know, explained a lot. And now, you know, now you see the problem of getting broadly neutralizing antibodies. You know, they have to get to the conserved parts of the protein surface underneath that huge panoply of blue glycans there. They've got to get, you know, into these holes and so on. And, you know, if you just looked at that, you'd think they'd never do it, but they do, as I said, if you look hard enough. And there are various characteristics of antibodies that do that. They usually need quite a lot of somatic hypermutation, and they often have long HCDR3s. But, you know, there are several different weaknesses where antibodies can get in. There's probably another one now, but these are the main targets, main sites that are recognized. I don't need to go into the details here. Here's an example from work that we did with Andrew Ward at Scripps from Cryo. And this is a broadly neutralizing antibody that binds. Here's the trimer. And the very long H3 goes into the near the apex of the trimer, which is kind of a bit unstable. It's got to blow apart when CD4 binds. Here's an antibody that's learned how to take advantage of this small weakness, gets in between the glycans, very long CDRH334 amino acids. And that's the kind of info you need to try for immunogen design. And as you'd expect, really, given, you know, all this sugar and the difficulties of getting in there, if you just immunize with this trimer in all these animals, you don't get broadly neutralizing antibodies. You get autologous nuke. You get antibodies that are directed to loops that are stuck out further in all of these animals. But one interesting thing that we noted, we did have some work with a guy called Vaughan Schmeider, who works on cow antibodies. And they have this weird long H3s. And so again, you know, this is just kind of like modeling in a way or whatever, but we just looked at this and we thought, well, you know, that's kind of interesting. Those long H3s could be made to get into some of these little places on the HIV envelope trimer. What if we immunize cows with this trimer? And we did that, and straight off, and the only time we've seen this, we got broadly neutralizing antibodies. And they neutralize quite well, and we've got one monoclonal that's just as good as anything a human's ever made, you know, searching for ages. And we published this, and we were worried actually because, you know, cows have a special place in India. We didn't know how this would be seen. But this was the response from an Indian source in Delhi. It's not actually that flattering when you read it. Holy cow, clueless scientists look for HIV cure in shock of their lives. All publicity is good publicity. I don't know if that's true, but anyway, that's what we did. Equally, or perhaps more significantly, was this little seen paper from Mitko Dimitrov that appeared in 2009. And what Mitko did was really quite smart and interesting. And what he did was he took some broadly neutralizing antibodies and he reverted them back as far as he could to their precursor or germline configuration. And then he asked, do they still bind to virus or HIV envelope? And they didn't. And so then you're faced with a question, and this is an absolutely general finding. So then you're faced with the question, well, how do these responses ever get going? You know, if the precursor forms of the antibody, that's on the BCRs, does not bind to these trimers, then how do you ever end up with a broadly neutralizing antibody? And the answer has to be it occurs in stages. And that's why it takes so long during natural infection. You need to manage this in stages. And in a vaccine, we'd like to manage that in these stages. And that's what we've kind of settled on. Indeed, the field has. They may call it different things. But basically, the notion is you need immunogens that set up, that prime the correct precursors. We call that germline targeting. You then shepherd the response along certain tracks, affinity maturation, and then you finish off with something that looks as close as you can get it to the native trimer on virions. And those are the kind of stages that we're trying, and many are trying to reproduce for HIV vaccine development. And this is, now I'm just going to not do it in much detail, just a couple of points. So Bill Sheaf, who I managed to recruit to Scripps probably eight or so years ago now from the U of W, he's been brilliant at this in designing germline targeting immunogens. And he does it by a lot of computational design, gets into a certain stage, and then he puts the molecules on yeast, in this case, or mammalian display in other cases, and selects with the appropriate antibodies until he gets a molecule that does what he wants, which is bind with as high affinity as possible to germline configurations of antibodies that look like the appropriate BCRs. And so this, you know, this is definitely not a molecule of natural infection. It's the outer domain of GP120 with CD4 binding site on it and a bunch of glycans put around to stop the antibody response going against the backside as much as possible. And so he takes that. Now that won't small, that won't be immunogenic. So he incorporates lumazine synthase into that, which is a self-oligomerizing protein. So it makes a nanoparticle. And it makes this nanoparticle, which is presenting multiple presentations of the CD4 binding site, you'll see. So the notion is to try and stimulate antibodies that can react against the CD4 binding site, particularly the VRCO1 class, so-called. And that is being shown to work very well. And what we initially did, and this was one of our contributions, so many years ago, we could see this kind of situation coming. This is me and David Namazi. So we could see this situation coming where you were going to design immunogens that were specifically targeted to human BCRs. So, you know, even macaque or monkey BCRs are a lot different to humans. And so what you would need would be human BCRs. So, but you can't do all this experimentation on humans. So how are you what are you gonna do? And so we came up with the idea of, and others did also follow, or around the same time, we came up with the idea of using knock-in mice and germline configuration of this antibody VRCO1 in a knock-in mouse. And this was then used with this molecule that I just shown you, EOD-GT8. And indeed, when the mice were immunized with this, they responded appropriately. And I don't have time to go into detail on this. Bill does a great job of describing this. So this gave us the confidence to put this molecule into humans, supported by the Gates Foundation. And that's actually gone in. The trial is finished. The results are there. They're not unblinded yet. Everything's been stopped by COVID, but hopefully you'll soon find out the results of this in the first germline targeting immunogen experiment, which I think is very important in terms of saying how much can we control human antibody responses by precisely designing our immunogens. It won't take us to broadly neutralizing antibodies. It'll just tell us that if we've succeeded in the first step, and you know, does this kind of shape design strategy work or not? Okay, and our lab also has contributed to this. One of the other factors that we like to understand as much as possible is, what do humans have in terms of a repertoire that we can prime by these methods? You know, what's the frequency of precursors and so on. And so we've done quite a lot of work, and I'm not going to read all this, but we've done quite a lot of work to try and understand the human repertoire. And actually, we think it's a lot bigger than what has been thought. So this is the accessible repertoire. So we think that humans can make not one human at one time, that's far in excess of the number of B cells, but the accessible human repertoire, we think, is about 1 billion, which is apparently, that number is a quintillion. And having all these sequences is actually quite useful. So I'll go back one slide onto HIV at the very end. I just have 2 quick slides on COVID, because once we were, you know, banished from the institute working on HIV, we assembled our team, and we've been working very hard to generate neutralizing antibodies to SARS-CoV-2. We started this about 7 weeks ago, and we've now got antibodies of, you know, really outstanding neutralization ability. And so what we did was, you know, and of course you've got to do all this from scratch. We actually established neutralization assays ourselves. We've also got them from others. We looked for the donors with the best newts, and actually SARS-CoV-2, people make good newts pretty quickly. Some do anyway, very quickly. Not like HIV that takes quite a long time, or even some other viruses, Lassa and so on. It's more like Hunin or something. They really bang the newt antibodies out quite quickly. We needed the proteins to characterize B cells, sort, We isolated about 1,000 monoclonals from 3 of the best donors. We found something monoclonals, and these are improving every day as we look more. And then we showed the most potent antibody completely protected Syrian hamsters from a challenge with SARS-CoV-2. This is in bioRxiv. We just stuck it there as soon as we'd finished, and we're trying to get it, into one of the regular journals. And just out of interest, I mean, my own take on the SARS-CoV-2 and COVID-19 is that one of these approaches is going to work, that we are going to get pretty good titers of neutralizing antibodies. I think the questions will be, are there any red flags when this is done? Because you might get great antibodies in 100 people, but if you get enhancement in the 101st, what do you do? And there was a lot of discussion and talk about possible enhancement in SARS-CoV-2. I don't think that's nailed at all, but it's a possible red flag. And the other thing is the longevity of the responses. If you have to vaccinate every few months, well, I mean, as it is now, it probably wouldn't be a problem, but long term, you don't want to have to be vaccinated every few months. So if there are complications like that, then I think there will be a need for further understanding of antibody responses and how to design vaccines to be more optimal. And that's what this review is about. It's about I think some of the lessons that we've learned in HIV applied to COVID if needed. And then the last slide I have is a rational approach to vaccines. This just basically summarizes everything we've done and are doing. And so just, as a takeaway, we started here, we got these broadly neutralizing antibodies. We had to work with those for a long time. Eventually, we began to understand how these antibodies recognize envelope. That started to lead us into immunogen design. We realized this was a huge immunological problem as well as one of structure and shape, if you like. And this led us to recruit I think some of the best people in B and TFH biology into thinking about this and coming up with better immunization strategies, better design of immunogens. And I haven't said nothing of this. And we also have tried to learn from natural infection, where a lot of lessons are available. We've fed all of these into design and strategies. This has led us into testing. And we've done a lot of that in particularly small animal models, also now macaques, and that's led us towards more testing in humans. We iterate a fair degree, for sure, especially in the animal models, and that we're hoping is going to lead us towards the ability to elicit broadly neutralizing antibodies through sequential vaccination against multiple sites, which I think it's a huge and very ambitious goal, but I think the only way to an HIV vaccine based on neutralizing antibodies. And I would just, and there's way too many collaborators to acknowledge all. I'll just say that, at Scripps, these individuals who are hopefully all mentioned have all made huge contributions. The Neutralizing Antibody Consortium, and more recently the CHAR and CHAR-VIDS. And, we've been very generously funded through the years by the NIH, by the Gates, by ARVI, and the Reagan Institute. And I'll stop there and take any questions. Thank you.
Thank you, Dennis. We have a limited capacity for people to ask questions directly. They can write questions in that I can read. Thank you for the inspiring talk. How many people did you need to screen to find 3 best donors for the SARS antibodies?
We've screened, I think we've got about 30 donors, but we focused on 17, and then we focused further on 3 out of 17. I actually don't think it's that difficult to find fairly, I mean, I've come from HIV where it's, extremely difficult, but I think for CoV-2, it's not so hard at all. And, providing you get reasonable donors, a lot of people are going to get pretty good NEUT antibodies.
I guess this would be a question asking you to think about the comparison between sequential immunization strategies versus gene therapy to deliver a broadly neutralizing antibody.
Yeah, I mean, I think gene therapy, I mean, I think AAV has suffered some setbacks, and, there have been a lot of antibody responses with AAV. So I'm not so sure about AAV. I think, like mRNA or DNA, mRNA may be a route. I mean, it's not, the levels have not been so great to date, but, they could be on the way up. And I think passive antibody, which I always thought was, kind of a bit crazy, especially for the developing world. If you put half-life extensions on there and your antibodies are extremely potent, it's not unreasonable to talk about antibodies that are good, that would give you protection for more than 6 months, even out to a year, maybe. Your costs are still way higher than, probably long-acting antiretrovirals at the moment. But there's a lot of effort going in to try and making antibodies much cheaper. And, that's possible as well. So I still think it's probably difficult for antibodies prophylactically to compete with long-acting antiretrovirals, but, maybe worth pursuing.
So here's a question that I actually don't know that this is true, but I don't keep up with things. Why do you think people are getting reinfected after they clear coronavirus?
I'm not sure they are. I think, I haven't seen the original papers, but my impression just from the media, and, I should read, actually I'm not sure the original papers are peer reviewed, but maybe they are. But my impression is that some of this was due, in the Korean case, was maybe due to falsely thinking that they'd be infected initially, so they weren't. And secondly, they may have RNA around, but do they actually have infectious virus around? Yeah, so.
Okay, so let me modify this next question just a little bit to broaden a little bit. What do you think the relationship is for neutralizing antibodies and the extent of symptoms?
I mean, from what we have done, I think the neutralizing antibody titers are a bit higher. I definitely don't want to write this in stone. This is totally, not statistically borne out, known. But I think our feeling is that maybe you have better NEUT titers if you've had a bit of a worse course. But I think, you need a lot more numbers to know if that's true. That, people who've had a very mild course maybe don't see as much antigen, they don't have as high titers. I don't think that the neutralizing antibodies per se are actually controlling the infection. I think maybe, but, it's because they do come up quite quickly, whereas in other instances, you can be pretty sure that by the time the infections, other viruses, by the time the infection's resolved, the NEUT titers have just started.
We'll get some more questions coming in, going to keep me busy. Is there a role for non-neutralizing antibody? That is, if you inject a non-NEUT antibody to GP120 into a macaque, does it have any therapeutic effect? It would be interesting to know whether functions like ADCC are playing a role.
That's quite a controversial area. I mean, as far as I know, no one has ever convincingly shown that by passive transfer that a non-neutralizing antibody can provide protection. I've never seen convincing data. there's quite a bit of correlative data where people have, used some sort of vaccine, immunogen, and found some level of protection and said there's no neutralizing antibody, it must be non-neutralizing antibody, which is not, which is a bit softer. And, maybe, of course, that's the RV144 Thai trial is supposedly non-neutralizing antibody. I don't know. I'd just like to see somebody put a non-neutralizing antibody in and clearly show protection in an animal model. And, I haven't seen that done.
I guess you could extend it and say you should be able to see escape then too, and some sequence change.
Yeah. Well, people did say that with regard to RV142, that there were pressure on the V2 region, but then there was pressure on all sorts of other regions that were not. So I don't know what that means.
Okay. What are your thoughts about the bi or multi-specific probably neutralizing antibodies, these engineered guys that have different specificities on different ends?
Tri-specifics and so on. I think very, very, very interesting. I mean, obviously you could produce them cheaper and you would have a lesser potential problem with neutralization escape. I think my worry would be with tri-specifics, that they would, because they look a bit odd, that they might have some kind of ADA response. But, according to Gary Nabel, I think that Sanofi's done a fair amount with tri-specifics, and I don't think they've seen problems to date. So, maybe.
Do you think probably neutralizing antibodies have potential as a functional cure via shock and kill strategy, or to indefinitely delay viral rebound?
Yeah, I mean, I think that's one of the, there are several groups and companies working along those lines, that if they combine broadly neutralizing antibodies with cellular immunity, with shock and kill, that they may, get control. So I think we wait and see what happens there, but it seems a reasonable possibility.
Why do you think many HIV vaccine studies in humans and non-human primates have found a limited association between protection and induction of neutralizing antibodies? These aren't my questions, I'm just reading them.
Well, I don't think that's true. I mean, the problem, well, I mean, there's two answers. Number one, you can't, the immunogens don't exist to elicit broadly neutralizing antibodies. So that wasn't possible. But we have published in the last 2 years, where we induced isolate-specific neutralizing antibodies, and then we challenged with the Shiv BG505, we immunized with BG505 SOCIT, and we got a perfect correlation between neutralization and protection. And we even did it on low-dose repeated challenge. So as the antibody concentration waned, you could see that the individual monkeys became infected once their antibody concentration fell below a certain level. We published that in Immunity, last year, I think. So I think that does say there's a correlation between induction of neutralizing antibodies and protection in macaques.
So a couple questions around, do you think the seasonal coronavirus has any impact on the type of immunization you get against SARS-2?
Well, I mean, you'd think it could, because at least there's some cross-reactivities there. But the neutralizing antibodies that we have seen, and I think others have seen, have very low somatic hypermutation. So it suggests that they came from a naive precursor, and they're not come from, a seasonal BCR that's well mutated to date.
So here's, I always tell students that favorite vaccine of the year, it's a pendulum that swings back and forth between neutralizing antibodies and T-cell vaccines. So let's see where you are on that pendulum right now. Do you have higher hopes for antibody-based or T-cell-based vaccines for HIV?
You know, I think that the best thing of all, which I've held pretty constant on this for years, would be that you induce strong functional responses in both arms. You know, so try and get I think if you have no neutralizing antibody response, it's very hard because, you know, before the T cell response kicks in, you know, the virus is going to multiply and it's going to be causing you all sorts of laying down its diversity. And, you know, it's just, so, you know, you could really do with, you know, both. So I would say, I mean, neutralizing antibodies alone, you know, you really need pretty good titers because you're looking for sterilizing immunity. You know, you can't let 10 virus particles get through. And so my answer would definitely be both. And I think that's what makes it hard also for antibodies for an HIV vaccine is, you know, I'm sure that your antibodies to measles are not, you know, sterilizing. In fact, there's evidence for that. You know, you have contact with measles, you can see a anamnestic response. You know, you don't get any symptoms because you've got enough antibody there, but I think so both. So let's try and have both. But you know, we just work on antibodies.
The kind of some overlap here in the questions that are coming. So let me close by asking a question myself. Do you know what the titers are in the AMP study? Did they reach this 1 to 400 titer?
The neutralizing titers. Actually, I'm not sure. What do I know of what titers they reached?
This is the passive antibody and
Yeah. And I mean, they give a really high dose. So, I mean, they certainly get those titers initially. I'm just not sure how far out I mean, they are re-injecting the folks fairly often. So I think they maintain pretty good titers, but I don't actually know the numbers.
Okay. So
yeah.
I think we'll let you go. Thank you so much for doing this. It's
my pleasure. Thanks everyone for listening.
Yeah, well, it's been fun for me to kind of interview you here. This is a fun experience. So thank you again, and good luck with your work.
Thanks everyone. Ciao.
Bye.